Good morning, everybody. This is Dr. Mercado from Sanoviv. I've been working in this majestic hospital for the last 8 years. And right now I'm in charge of the IV therapies and medical programs and also working as a chief medical advisor. The topic we're going to talk about today is predictive biomarkers. Basically, this is about preventive medicine, the cheapest way to go about medicine. So, today we can identify problems before they become serious. As soon as the body is simply out of balance, before actual damage has been done. Each predictive biomarker is selected by its sensitivity and specificity. That is, its accuracy and its lack of false results. So, what would happen if you could prevent a disease by some simple blood tests? Predictive biomarker test results provide a comprehensive, accessible, actionable, and personalized plan for health. Biomarkers are key molecular or cellular events that link a specific environmental exposure to a health outcome. They are typically measured in readily available body fluids, blood or urine. They are able to detect subclinical disease, are used to monitor clinical and subclinical disease burden, and to monitor also response to treatment. The 8 biomarkers we're going to be talking about today are here on the screen: hemoglobin A1C, C-reactive protein, homocysteine, oxidized low-density lipoprotein, or known as LDL cholesterol, 8-oxoguanine, vitamin D, food sensitivities, and fasting insulin levels. So let's start with our first biomarker, the first blood test that you can check to understand where you're at. Hemoglobin A1C is produced by a non-enzymatic glycosylation, meaning that this reaction, this attachment of sugar, blood sugar, to the hemoglobin does not require any cofactor, does not require any enzyme to produce this reaction. So it links or it binds to a chain, the beta chain of hemoglobin. Since this reaction is controlled by mass action, meaning the volume of blood sugar, and the red cell is permeable to this blood sugar, it just lets it in, the quality of hemoglobin A1C formed is directly proportional to the average plasma glucose concentration during the red cell's 120-day lifespan, which means that this assay can let us know the average blood sugar levels in the last 3 to 4 months, which is the half-life of each red blood cell. A hemoglobin A1C below 5% indicates that the body is effectively managing blood sugar levels. When levels are elevated above 5%, that means the body is no longer properly managing blood sugar levels. Elevated levels also reflect a tendency towards higher inflammation throughout the body. Current guidelines identify an hemoglobin A1C level between 5.7% and 6.4% as an indicator of pre-diabetes, while levels of 6.5% or more, it's a criteria for diagnosing type 2 diabetes. So our goal value of this first biomarker, hemoglobin A1C, when you check it in your blood, we want to keep it below 5%. Our next biomarker, fasting insulin, it's also a blood test. Insulin helps sugar move from the blood into the cells, where it can be used or stored. Chronically elevated blood glucose leads to insulin resistance and numerous chronic inflammatory diseases, including diabetes and heart disease. In this diagram, this graphic, we can see the insulin molecule binding to its receptor on the cell membrane and triggering all the signal pathway that opens the gates for the sugar molecules to enter the cell. Insulin and insulin-like growth factors are well known as key regulators of energy metabolism and growth. There is now considerable evidence that insulin and the IGF signaling may also be relevant to dietary and lifestyle factors that influence cancer risk and cancer prognosis. Soda, candy, pasta, bread, pastries, and fruit juice are examples of foods that quickly break down into glucose once consumed, generating a spike in the blood glucose level and the corresponding release of insulin. It is becoming very clear that longevity is intimately tied to this cycle, and by modifying your diet to restrict sugars and grains, you can slow down the rate at which your biological clock is ticking. Our predictive goal value, when we take this blood test, we want to see fasting insulin levels below 3 micro international units per milliliter. Now we're going to move to our next biomarker, which is called high sensitivity C-reactive protein, which is a protein produced in the liver. As a predictive biomarker, high sensitivity C-reactive protein reflects the effectiveness and efficiency of immune defenses, which are responsible for neutralizing any sign of infection, repairing daily wear and tear, and identifying and eliminating cancer cells. When C-reactive protein is high, that is a definite indication of an inflammatory process present throughout the body, even though in some cases inflammation is present, yet CRP is not elevated. High C-reactive protein levels are common in pre-diabetes and diabetes, reflecting insulin resistance and metabolic syndrome, all conditions associated with a high risk of cardiovascular disease. Elevated CRP levels can also indicate a long-term infection. The predictive goal value of this biomarker is ideally to keep it below 0.5 milligrams per liter. Very well. The next biomarker we're going to talk about is vitamin D. Sunlight is required to synthesize vitamin D from a cholesterol molecule precursor that is in the skin and then continues its transformation in the liver, and finally the active form of vitamin D is made in the kidney. Vitamin D moderates cell division. It provides vital communication links between cells to normalize cell growth and prevent uncontrolled cell production. Vitamin D also prevents autoimmune disorders. Control mechanisms are built into the immune system that prevent immune activity from causing damage due to inflammation. Vitamin D is essential to the function of these control mechanisms. It regulates many genes that play a role on these mechanisms. Vitamin D also reduces inflammation in the brain and nervous system. Vitamin D moderates the inflammatory processes in the neurological system. This is particularly important since the brain lacks the regulatory systems to moderate inflammation effectively. In addition, according to the National Institute of Health, vitamin D may play a role in prevention and treatment of type 1 and type 2 diabetes, hypertension, glucose intolerance, multiple sclerosis, and other medical conditions. Our optimal level of vitamin D is between 50 to 70 nanograms per milliliter. Our next biomarker, it's called 8-oxoguanine. The main consequence of oxidative stress is the formation of DNA lesions, which can result in genomic instability and lead to cell death, premature cell death. Guanine is the base that is most susceptible to oxidation due to its low redox potential. This means its low ability to neutralize free radicals. So 8-oxoguanine is the most common lesion. These characteristics make 8-oxoguanine a good cellular biomarker to indicate the extent of oxidative stress. When antioxidant levels are sufficient, it prevents oxidative damage caused by free radicals. Healthy levels of antioxidants such as vitamin C mean efficient energy production in the cells. Elevated levels of both 8-oxoguanine and oxidized LDL indicate higher levels of inflammation and oxidative stress throughout the body, factors that underline almost any form of chronic illness. This takes us to our next biomarker, which is oxidized LDL cholesterol. And it's important to differentiate because native non-oxidized LDL cholesterol does not cause atherosclerosis, heart disease, or stroke. LDL is only bad or only causes disease when it's damaged or oxidized. Only when LDL cholesterol is oxidized, it leads to inflammation in the arterial lining and leads to arterial plaque, no matter what the level. The higher the level of LDL cholesterol, of oxidized LDL cholesterol, the more likely it is to be oxidized, particularly if you are not taking a full spectrum of antioxidants. eating right, not smoking, and not eating fried foods, particularly trans fats. The predictive value for this marker, no matter what age, it's to keep it below 60 to be on a low risk of developing arterial damage. So, the best treatment for oxidized LDL cholesterol is not statin drugs, but antioxidants. Our next biomarker that can be checked in the blood is homocysteine. This chemical byproduct of cell function is a widely recognized biomarker of cardiovascular disease and risk, and looks at risk independently of other markers like cholesterol. Homocysteine levels reflect a process that occurs within every cell in the body, known as methylation chemistry. Methylation supplies methyl groups for a large number of biochemical reactions in the body. It controls the overall sulfur metabolism, balancing the needs for methyl groups for glutathione synthase to control oxidative stress. So this methylation is vital to keep normal glutathione levels within the cell. It also coordinates the production of new DNA with the supply of methyl groups, which are used to methylate DNA, among many other roles. So when this process is not working properly, homocysteine levels are elevated therefore. This marker reveals function or dysfunction on the most basic level. Elevated levels of homocysteine cause damage throughout several mechanisms, including the generation of reactive oxygen species, hypomethylation, and including endothelial dysfunction, vascular inflammation, and neurological diseases like Alzheimer's and Parkinson's disease. The predictive goal value for this biomarker can be checked in the blood. It's 3 to 6 micromole per liter. And our last biomarker that can be also checked in the blood are the so-called hidden or delayed food allergies or sensitivities. Delayed food allergies are very common and often go undiagnosed. As delayed food allergies are associated with a different type of immune response, it's IgG mediated. It's not an immediate response like the IgE. Well, like some people react immediately to a bee sting or to peanuts. This kind of reaction causes a relatively subtle symptoms over days or weeks. That's why it's difficult to really associate which food is causing the symptoms. Symptoms can include gastrointestinal problems like constipation, diarrhea, gas, cramping, and bloating, and also rashes and headaches. They might play a role, these kind of delay food allergies or sensitivities can play a role in many diseases by the burden they create in the immune system. These diseases include asthma, attention deficit disorder, rheumatoid arthritis, canker sores, ear infections, eczema, irritable bowel syndrome, migraine headaches, psoriasis, ulcerative colitis, and Crohn's disease. It is common to see foods such as gluten, found in many grains such as wheat, barley, and rye, dairy, eggs, and soy to cause delayed food allergies. These are the most common allergens in food. For example, someone might eat a sandwich with wheat bread, then a day later have digestive upset. To give another example, someone might eat some cheese for dinner and wake up in the morning with a stuffy nose or constant sneezing. These are both examples of delayed food allergies. So, the more food intolerances or allergies an individual has, the higher the likelihood of developing leaky gut, augmented intestinal permeability, and the bigger burden these create on the immune system surveillance. And this can trigger an autoimmune disorder. So when we run this test, what we want to see is no delayed allergies. In summary, these are the 8 blood and 1 urine tests that we run at Sanoviv and can work as a predictive biomarker. It's part of a checkup, at least a yearly checkup, to know where you're at. The values described on the right column are quite not easy to find actually with our current diets. Nevertheless, it's important, and we encourage you to come to Sanoviv to get a thorough checkup. This is just a little part of what we do on the medical department here at Sanoviv. Nevertheless, you will not only get these blood tests, but you will get the assessment of the dentist, nutritionist, chiropractor, psychologist. And that really creates an integrative approach to understand where you're at and what is the plan, the tailored, customized, personalized plan of action for the next 6 months in order to prevent, which is the best way to go about health. An ounce of prevention is worth a pound of cure. So we invite you to come to Sanoviv and experience the difference on medical treatment. Thank you everybody for listening, and if there is any questions, we are here to answer them. All right, well, see, there are no questions popping up. So with this, we close this presentation, and we thank you again to whoever listened to this one.